in problems. And I think what he's accomplished a lot. I think it's very impressive, and I'm really going to miss having someone that's become an expert in this important group to talk to and bounce ideas off of. Now that Juan is finishing up and moving on to other things, so there's a lot more I could say about Juan. Many of you sort of know him, but I'm just going to let him tell you about his research project. So, thank you.
Well, thank you everyone for being here. I hope the public that we have online can hear me well and everything goes right in the presentation. I'm going to try to summarize the work I have been doing during the last 7 years on this group of flies. The title of my dissertation is Systematic Taxonomy and Ecology of Tachinids, focus on the Polideini, but I have summarized this to the neotropical Polideini because this is a small group within the whole family that I have been working on. So I'm going to start with a broad introduction to the topic to provide some context, and then go through the 2 chapters of my dissertation. That is first a phylogenomic reconstruction of the tribe Polideini using ultra-conserved elements, which is a next-generation sequencing approach. And then my second chapter, which is a partial taxonomic revision of the highly diverse genus Chrysotachina. First, I would like to give acknowledgements to my committee, especially to my advisor, John Starman, who I have had constant discussions that have helped me to develop my project, to the late Monty Wood, who I had the opportunity to meet for a brief time and who shared with me a lot of his knowledge. A mi papá, mi mamá, hermanos, mi sobrina, mi cuñada, amigos, amigas, friends, parceros y parceras, and to the biological, to the right state community also in general. Thanks for all your help. And to the many people who has helped me a lot on this long process, to all the curators in the collections also. So first, flies. When we think about flies, everybody just thinks about the house flies, the mosquitoes. But this is really one of the most diverse groups of organisms in the planet. 1 out of every 10 animals, animal species in the planet is a fly in the sense of the group. All flies have 2 pairs of wings, 1 pair of wings. And that's what defines the whole group. And there are over 160,000 species in the world. They are divided in 163 extant families in the planet. And my research focuses on a single family, which is the family Tachinidae. Flies have diversified not only in numbers, but also in habits. They have adapted to colonize all kinds of environments and niches. There are groups of flies that have developed habits as plant feeders or fresh material. And they are in all kinds of environments. They are in diversified highly in aquatic environments. Some lineages are predators. So other ones have developed blood-sucking habits. Other ones are decomposers, parasites, pollinators, and the group that I work with, parasitoids, which also are pollinators. So what is a parasitoid? The parasitoids are organisms that develop their life cycle relying on other organisms which are their hosts. They usually lay their eggs on their hosts. These species develop in their immature stage in or out their host and complete their life cycle, and eventually they kill their hosts. In the video, you could see a female tachinid, which is laying its egg on the caterpillar. And in this case, that egg is going to emerge, and then it's going to eat the caterpillar from the inside. But this is not only a single group of organisms that are parasitoids. There are many groups of insects. Most of them belong to Hymenoptera, the order Hymenoptera, where we have wasps, and Diptera, where we have flies. And their most common hosts are slug snails and most of the major terrestrial arthropod orders. It's important to study parasitoids because they are the dominant natural enemies of phytophagous insects, and they help us to understand community structures and how the coevolutionary and evolutionary diversification has occurred in the planet. We don't have any parasitoids as humans, but like the closest thing that humans have imagined can refer to the movie Alien. And this is basically what happens to an arthropod that is being parasitized. It's being eaten from inside the flesh, and then there is this chest burst when their parasitoid emerges to try to complete their cycle. And then the host dies. So the family Tachinidae is highly diverse in terms of numbers. It has over 9,000 described species, but this number could be at least double that number of species in terms of undescribed species. It's cosmopolitan, is present across the world in all major continental land areas, and all of them are characterized by their obligated endoparasitoid habit. All of them are associated, or at least from what we know, all the species are associated with hosts which they parasitize. A bit of introduction to the relationships of this family. This family belongs to the superfamily Oestroidea, and this is a phylogenetic reconstruction of a paper that I collaborated during my PhD, using also the same methodology that I used for my first chapter, in which we did a phylogenetic reconstruction of the whole superfamily Oestroidea, and the families that are related to Tachinidae are also families that have developed, some of them have developed parasitic habits, like the Polideini, for example, which is sister to the Tachinidae. This group parasitizes earthworms, and there are other groups like the rhinophorids, they parasitize pillbugs, and there are some other groups that are more scattered across the tree, which also some parasitoid habits. The family is divided in 4 subfamilies, but each one of these subfamilies has many tribes within. We are talking about almost 60, 55 tribes. Okay, sorry for that. We had some issue with the batteries, but I hope that you can hear me online again. Okay. So I was describing the diversity of the whole group Tachinidae, and this group is poorly known and taxonomically difficult. It has a lot of morphological diversity, which recurrent convergence, which turns out in many distant groups that have similar morphologies, and even in the male terminalia also a lot of convergence that difficulties the classification of the group. The relationships among the tribes and the most of the genera are poorly understood, particularly in those 2 families, subfamilies, sorry, the Tachinidae and the Dexiini. Those are the families represented by green and red in this phylogenetic tree. And it's a bit small, but you can see the triangles are showing the taxa, or the groups, in this case, the tribes that have been recovered as paraphyletic in this tree. So we still have a lot of taxonomic work ahead to solve the relationships of the tribes and most genera for the Tachinidae. One of the main problems that we face in the taxonomy of this group in the neotropical region is that This group is a very poorly studied. It's one of it's found in the tropics. Is the most diverse among most of the biomes in the planet. But most of the species have been described over 100 years ago, and most of the species are known from very short and brief, uninformative descriptions, and those species are usually known from a single holotype specimen. And also, for most of the neotropical species, we don't know anything about the biology. Now, regarding the Polideini, this tribe belongs to the Tachinini. The group was just redefined around 20 years ago by O'Hara. And in the classification of this group, he focused on the Nearctic diversity of this group, and he put together in the Polideini taxa that were assigned before to around 9 tribes belonging to different subfamilies, and this is because of the broad diversity, morphological diversity and disparity of the group. The group is defined based on a combination of 9 characters, 2 external characters, and 7 male terminalia characters. In their external morphology, the Polideini can be recognized by the presence of the long and ephemeral seta, where you can see here by the red arrow. And in the square, you can see the posterior spiracle or the metathoracic spiracle, which in the Polideini has these 2 lappets that have almost the same size, forming this V shape. The combination of those 2 characters, the combination of these 2 characters with the male terminalia, is what gives the composition for the diagnostic of the Polideines. This group is mostly diverse through the New World, but there are 6 genera that occur in the Palearctic region. From these 6 genera, there are 3 that are exclusive to the Palearctic region, although I don't know the relationships of those 3 genera. Those were not examined in my study. There are 3 genera that are shared between the Palearctic and the New World, and those 3 genera that are shared with the Palearctic region are also present through the Neotropical region. The fauna of the Polideini is better known in the Nearctics. There are 15 genera that occur in this region. 10 of those are shared within the tropical region, and the group is most diverse through the Neotropics, and particularly through the Andes region. This is a group that has surprisingly diversified through all elevations in the Neotropical region, especially in the Andes, from the level of the sea up to 4,500 meters, which is, I think, close to 20,000 feet, something like that. So this is a group that is very interesting within the Tachinidae, because it's a group that it's actively radiating, and the whole Tachinidae is a family that is that has a fast diversification. But there are several clades that have been characterized by some shifts in diversification rates, and the Polideini is one of those. And this has translated to a lot of convergence in their morphological shapes, and also in an abrupt diversity of host associations. So with that in mind, I'm going to start presenting the first chapter of my dissertation. There have been several works done on the phylogenetics of the Tachinidae. However, all the previous works that have been done on tachinids are based on single sequencing, where the phylogenies are recovered with lower support for the relationships. And also, most of these works have been done based on a small taxa representation, which limits the power of this analysis. The last work by Starman included around 500 taxa, and it's providing a good framework for the understanding of the diversification of the family. In that work, there were 11 taxa included for the Polideini. However, only these three basal, these three basal taxa are neotropical. So most of the neotropical taxa are poorly known. In that work, it's remarkable that the Panseria, that is a genus that belongs to the Ernestini, was recovered as sister to the Polideini. And I'm going to talk about that more in a bit. So the next generation sequences provide more support and more robust support to our phylogenetic analysis. These allow the capture of large target regions in a single experiment, which make this saves costs in the sequencing of these experiments. And there are different strategies that have been developed, and I have used an ultra-conserved elements approach, which is based on the hybridization-based strategies. In the ultra-conserved elements approach, these highly conserved informative regions in the DNA across broad groups in the phylogenetic tree are recovered and are sequencing to compare their diversification. So over here we can see an example of groups that are highly diverse, are highly distant, anoles, a zebra fringe, and a chicken. And in those groups, there are these ultra-conserved elements that have been preserved and can be used to compare these distant groups. And the longest the distance of the base pairs on the DNA from the core of the UCE, the mode variation, and that's what is used to infer the phylogenetic relationships of these groups. The UCEs have been started to be implemented in the study of the phylogenetics of many groups of insects. Here we have an example of weevils, of Australian small weevils, the phylogeny of the Ostroidea that I was co-author in. But there are many other projects that have started to use these next approaches in resolving phylogenetics of many fly groups. For the methods of this part, I conducted the wet lab experiments, all the DNA work at the lab at the Smithsonian Museum in 2018. I started with an initial data set of 62 Polideini taxa. And we used, or I used samples that were stored in alcohol, which are the traditional approach. But one of the advantages of using next generation sequencing approaches is that those allow us to use pin museum specimens, which might have some degraded DNA, but because we are targeting hundreds of chunks of the DNA, we can still recover some of those that can be used for some of those chunks of degraded DNA, sorry, to include these samples in our phylogenetic analysis. The experiment was run with a probe design for targeting calyptrate flies, which is something that it's hard to find, but I luckily had this other project led by Eliana Buenaventura, and she was working on designing this project for solving the phylogeny of the superfamily. So I was able to use the same probe set for my studies. This probe set is targeting over 2,500 loci, and it was used running the high-sec Illumina sequencing technologies. For the bioinformatic analysis, I cannot give details now, but it's run with the Phyluci package, which is a series of scripts for doing all the cleaning of the data of the sequences. And for the phylogenetics analysis, I run a RAXML analysis. And I evaluated different data sets with different amounts of coverage and identity to assess the stability of the taxa in the reconstructed phylogenetic tree. This is just a visual representation of the wet lab part. So the DNA is sheared in the small parts that that we can recover the UCs. The probe sets make that the targeted DNA sticks to them, to the probe set. All the DNA that we are not targeting is washed away, and then we send everything in one sample to be sequenced for the posterior bioinformatic analysis. And so based on these experiments, I was able to reconstruct the phylogeny of the Polideini with high support for most of the nodes. From the 62 samples that from the 62 taxa that were that I started the experiment, I recovered the tree with high confidence for 59 Polideini taxa, and the taxa cluster in 11 major clades. So this is the first finding of the tree. It includes most of the Polideini taxa. Some of these nodes were recovered with bootstrap support lower than 95%, but still most of the relationships within and between major clades were recovered with the high confidence. And this very vertical structure with very short internodes suggests rapid diversification along the whole tree, along the whole tribe. So now, based on the distribution of the taxa that I included in the experiment, I color code the different branches based on their distribution to see if I could find any patterns in the distribution. First, the most basal groups in the Polideini, those have a neotropical distribution, which suggests that probably this group originated in the neotropical region. However, this cannot be ascertained because we don't have any fossil records to confirm this hypothesis. But also the Panseria genus, which is sister to the whole group, has an Nearctic distribution, which is kind of contradictory, but also at the same time, we have to keep in mind that the neotropical Polideini, the tachinids in general, are poorly known. So we need to do more explorations to understand this. Other major findings about this is that there might have occurred multiple migrations between the neotropical and the Nearctic region, possibly with several reverse migrations. There was only a single lineage that was recovered mostly composed by Nearctic taxa in North America. And if we can see here, these two, Spilacatosoma and Maromaya, those two taxa had also species that are present in the neotropic region. And those are the ones that could have the reverse migration. There are 3 taxa that independently, they might have colonized the Palearctic region, and those were recovered in highly derived positions within the tree. So there are several factors that may have affected favor the diversification of the Polideini, and one of them is the flickering connectivity in the Andean mountains, promoted by the glacier interglacial cycles. We might have favored the connection between high peaks taxa that was diversifying in these high mountains, and then associated with these glacial interglacial cycles have spread to colonize other regions. And also the colonization of novel hosts might have favored the diversification of this group. Unfortunately, the host records that we have for the family are very low. We have poor data for the host records, but still we can infer some conclusions of the diversification of this group based on their hosts. First, probably the ancestral last common ancestor of the Polideini was probably a caterpillar. The sister group of the Polideini, Panseria, parasitizes caterpillars. But through the tree, we can see different host shifts to other groups of insects. There are host shifts to crickets in the Orthoptera, the scorpions, to centipedes in the Chilopoda, and the Blotteria, which are the cockroaches. And all these host shifts are independent, have occurred independently. So now, assessing the diversity of the group, I included here just a small representation of 1 or 2 representatives for each one of the 11 major clades that we have in the whole tribe. And when we see this whole amount of diversity in the group, it's hard to find patterns in the morphology of the lineages that belong to this farm and to this tribe. And from here, we can see that the Polideini kind of reflect the whole diversity found across the whole family. Even close related taxa, it's hard to find morphological patterns that can allow us to recognize them. Now, if we see this is the male terminalia of these representatives, we find the same patterns of diversity of the structures. Maybe on the epandrium, which is this capsule that has these claspers in the genitalia of these flies, we can see that these groups, the K, H, and J, have a more pointed tip dorsally, and the more derived might have a more flat dorsally surface. But however, I have found many exceptions to these cases in both parts of the tree, in the more derived branches and the more basal branches. And I have put this together just to show a more deeper representation just within part of the tree. In the clades A and B, for example, if we go and start sampling multiple species within this clade that belong to different genera, it's hard to find patterns, morphological patterns that allow us to associate them or find the relationships based on the adult morphology or on the male terminalia. And just what I was saying about the shapes on the male terminalia, for example, that on the more derived groups, we can find that epandrium capsule that is more flat and more rounded. And this is smaller Syncercus, which is this small structure in the middle. This tends to be smaller in those groups. But if we see this one, it's closer to the structure that we can find in the more basal groups. There are many patterns in the forms and shapes of these groups. And I'm going to go through several of them. The first one is what I'm calling the hedgehog flying syndrome. And this is this convergence to this body shape in which many unrelated taxa of the tachinids have come to these more globose, robust, spiny shapes. And those occur across the tachinid tree in other also unrelated groups of the Tachinidae. And that occurs mostly through the Andean region and mountainous regions of Central America. Most of the taxa that has diversified with this shape occurs in this region. So now if we see more groups of the Polideini, for example, we can see that at least 5 times this body habitus has arisen within the tribe independently. And there are even other taxa that I couldn't include in this phylogenetic analysis that I haven't been able to associate with other groups based on morphology. So for this, future studies need to include these other taxa to comprehend better their associations, if those could be other potentially independent forms that have evolved with this body shape. And this body form also, in some cases, has evolved not only in shape, but also in color. But still, we can find the convergence to these shapes, but there is nothing known about the biology of these groups. Other convergence patterns, we can see this yellow coloration with this black stripe on the abdomen has evolved at least twice within the tribe. And I have seen also flies belonging to other clades of the Tachinidae that also present this same pattern in the Andean region. The metallic color, for example, was thought that only had arisen within the genus Chrysotachina in this lineage age. But I have found specimens, female specimens, that I think probably are close to this lineage F that also exhibit this metallic coloration. But unfortunately, I cannot confirm those relationships because this is not only for 2 or 3 females, and I don't have male terminalia characters to suggest any associations with other clades. So these are poorly collected groups. In the body coloration, there is also a confusing convergence in terms of some microtomentose patterns on the front orbital plate, which is here this top part of the head, and also in the lower part, this is the gina and the vibrisa insertion. In those parts, there is these dark markings that occur in several groups across the tree, at least 5 times. I have characterized that within the tribe. And this is another particularly cool example because these are specimens that belong to 3 different clades of the Polideini, and all of them are known from the same place in Santa Catarina in South Brazil, and all of them converge to this same body color with the same body pattern, same microtomentose pattern on the thorax and head coloration. Why this is occurring, it's completely unknown, but these are co-evolving in the same space, converging to the same body pattern. The convergence is not only just restricted to external body color. There is also convergence in structures. There is this outgrowth of abdominal sternite ceti, which are structures on the lower part of the abdomen of the fly. And these outgrowths that look kind of combi of ceti have occurred have evolved independently multiple times within the Polideini, but I also have characterized that outside the tribe. So there are many other groups that could, closely related groups that could also have and present these patches of ceti. What are they for? We don't know. Those should be involved in copulation because those are also only found in males. But we don't know anything about that. So to conclude this chapter, the Polideini have experienced a recent and explosive radiation, which has turned in a lot of convergent forms in their external and in their male terminalia. The great variation that is displayed by the Polideini is also found within their clades, which makes difficult to characterize their genera, and also makes a lot generates a lot of forms within each one of these genus closely with similarities of other groups. And the group probably diversified in the neotropical region with several colonizations between the north and southern hemisphere in the New World, and a few colonizations to the Palearctic. Probably the ancestral host of the Polideini was caterpillars. And also the delimitation of many genera in the Polideini is limited by the low amount of specimens collected or found in collections. I have on loan specimens that come from the main collections in the Americas, from Brazil to here in the United States. I have specimens from the British Museum. So these are the museums that have the broadest of the depositories of this family of this, on the whole Tachinidae. And still, for many genera, I have only a few representatives or only one specimen that could be a male or a female. And when I only have one single, I cannot examine the male terminalia, which is the most diagnostic for the morphological taxonomy of this group. And we need to do intensive collecting for these groups, which probably many of the flies I'm working with are probably already extincted out in the nature. So the second part of my work is a partial revision of the genus Chrysotachina, which is, this work is based on morphology mostly. And this is the clade that was recovered in this major clade age within the Polideini. And we can see some patterns that I'm gonna start pointing out one by one. This is a group that is highly diversified to the New World from the southern part of Argentina, from the southern part of Brazil and northern part of Argentina to South Canada. This genus is characterized by a diversity of forms. So we have from these small body forms, which are close to 5 millimeters, to the large body forms that have close to 15 millimeters. And there are 80 species known, and 72 species are treated in my work. This is a group that is a good example of the striking similarities exhibited by the group and the disparities also present in the Polideini. The concept of the group has evolved slowly. Before were considered only metallic species, and Guimaraes in his catalog, like 50 years ago, listed 13 species. 2 of those were non-metallic. Then Nunes contributed with 7 new metallic species, but still that was the main criteria to define what was belonging to this tribe, to this genus. Later, O'Hara expanded the concept of the genus, including more species that have been already described, were non-metallic, but were considered as species in other genera. So he broke them to the Tachinidae, to the Chrysotachina, and he described 3 new Nearctic species. Previous to my study, there were 29 described species. This group is highly diverse in hosts also. Luckily, I had access to records of 34 species of Chrysotachina, and these Tachinids parasitize caterpillars of 4 families. Most of them are shelter builders, and there are some species that are also free-living caterpillars. The host records or the specimens that have been reared also offer the opportunity to explore the morphology of the puparium, which is the case that is left from the metamorphosis between the larvae and the adult of the fly. And this structure also is of taxonomic value and offers some characters that can be explored to understand the diversification of the group. So from this study and the previous study, the specimens were borrowed from 16 museum collections. I had access to close to 700 specimen records reared from caterpillars, mostly from 2 initiatives, one Guanacaste in Costa Rica and Yanayacu in Ecuador. But outside these 2 initiatives, the knowledge of or the availability of reared records is almost 0. The characterization of all the species was based on morphology and male terminalia, and I also had a sequence available for 255 specimens, which correspond to 65 species. And these barcodes also were used to define the species concepts defined for my group. So based on an integrative approach using the phylogenomic data, the CO1, which is the barcode data, the adult morphology, the puparium, the host records, well, puparium when available and host record when available. I redefined the genus, described the species, and proposed 9 species groups for the genus. And additionally, I generated a dichotomous key following a format with the text description, but also with illustrated pictures of the external morphology and adult and male terminal morphology for their identification. So the ultra-conserved elements phylogenomic reconstruction provide a framework for the understanding of the whole genus. Because first it was considering only metallic and non-metallic forms that were similarly looking in the external morphology. But first it was recovered this Chrysotechina sp49, or which belongs to this Arangoi species group. This is a group that was completely that wasn't known before, and this is a group of forms of Chrysotechina that have a slender body. At the beginning, I was associating this with a new genus, but once I started to put all the dots together and know more about the host records, for example, this one is known from a free-living Hesperid, Nymphallidae, and the morphology of their puparium. I included this in this genus, also well supported on this data. Also based on this reconstruction, some of the non-metallic forms seem to be more basal inside the genus, reconstructed a sister of the metallic forms of Chrysotechina, and the forms of Chrysotechina with this robust and spiny body shape also have were recovered twice independently inside the tree, suggesting that these body forms have evolved independently within the genus. The same form, this kind of like the same way as it has occurred in the tribe. So based on the 65 records of the CO1 gene for the whole genus, I was able to build this tree in which the relationships cannot be trusted due to the low support given by the barcode, but this provides a good reference that again mixed with the morphological and life history traits help me to define the 9 groups of species and the delimitation for each one of these species concepts. For example, this species group, which is called Ornata, is grouped by a similar morphology, and all of these, the known records for this species group, these species parasitize caterpillars of the Hesperids that feed exclusively on monocotyledonous plants. So there could be a co-evolutionary process, and these are the first basis to keep exploring the diversification of this group. So some aspects of the diversification of these, the Chrysotechina, the more basal branches are the flies or the species that have a slender body habitus. The hedgehog body habitus, based on CO1, it was recovered independently 6 times, but again, we cannot draw these relationships. Based on morphology, I proposed that this body habitus has evolved at least 3 times independently. And the typical body habitus, in which we have the metallic and non-metallic forms, it's distributed across the whole group. In terms of their host use, most of the known species of the Chrysotechina parasitize shelter-building caterpillars, but the most basal group over here, the Arangoi species group, parasitized nymphalid species that are free-living caterpillars. The other records that we have on nymphalids, this green color over here, are the shelter builders. So it seems like Tachina shifted from free-living caterpillars to shelter builders. And we have at least one species that has shifted to parasitize radionidae caterpillars, which are caterpillars attended by ants. That's all what I can say based on only on the records. We don't really know more about the diversification and host use of the Chrysotachina. Okay. Also, the puparium is important for this group. All the Chrysotachina have the posterior spiracles, which are kind of the respiratory structures of the pupa. They have this convoluted shape, like this reticulate shape on their posterior spiracle. All the non-Chrysotachina, all the other Polideini taxa I found, they have these slits with this shape. And also within the genus, I have characterized 3 different types of these these spiracle shapes that also help to diagnose and understand better the diversification of the group. The sexual patch is a structure that is present on the males ventrally. This is this agglomeration of hairs, and it has different configurations with different depths on the cuticle of the structure. In some of them, there is some outgrowth of pile-like microtomentum. But this is something that is not exclusive of metallic species, which has probably, sorry, it has evolved multiple times also probably within the genus. And lastly, a broad recognition for the Chrysotachina. So there are several things that we should keep in mind to recognize the group. Some of them are characters about how diverse they are in terms of shape, in terms of color. But there are also some other characters that are common to other groups, but we need to keep to account to diagnose, to recognize this group. The presence of these, for example, these 2 seta on the dorsum of the thorax have been considered diagnostic for this group, but they are also present in many other genera. So we need to do an intricate list of characters to recognize the Chrysotachina of what they possess and what they don't possess also. Because also the tribe in general is still poorly known, and there are many undescribed taxa. So there are many other genera that haven't been described, but they exhibit similar forms like Chrysotachina, and they have characters that are not found within Chrysotachina. Like, for example, these other species that they have those dark markings. Chrysotachina doesn't have that. Also, we have metallic species in Chrysotachina. There are other undescribed species of Polideines that in this case, for example, they have this laterocline city on top of the head on the vertex. That's something that is absent on the Chrysotachina. Chrysotachina is also characterized by, there is in the back a row of situli that goes on the top of the head of the specimens. In Chrysotachina, that's interrupted here medially. In this other undescribed taxon, it's continuous. So that's another factor that needs to be kept in mind for the recognition of the genus. On the genus, there are these small setae that grow here on the anterior ventral part. In other undescribed taxa of the Polideini, these setae have a stronger growth in the more dorsal part of this portion, which is kind of like the cheek of the fly head. And additionally, the face is kind of protruding, projected. And there are 6 additional characters. that are necessary to be expected to confirm which is a Tachina. So as a summary for the genus revision, the genus concept needs to be expanded to include bodies with different body shapes, body habitus, several species await description. And the same as with the Polideini, we need to do a broader sampling of these flies to know which is their true diversity. I have characterized 80 species or identified that there are 80 known species, but the number of species, undescribed species for sure, is a lot higher. And that's it. So yeah, questions here first on the audience here, and then I'm gonna pass to the WebEx to get questions. Okay, so the question is about this convergence pattern. I think that this converging pattern is a derived trait for each one of these different three species, because at least in the Tachinidae, which is the one I have explored more in detail, the more basal species in this group have a different body shape and a different body color. Why they are converging to this shape? That could be associated with living in the same space, and actually I'm not sure if they are mimicking each other or if they could even be mimicking other groups. Like there's the flesh flies, which also present often similar colorations. So it's hard to know who is copying who in these cases. You're welcome. Any other question? Well, I haven't seen any patterns of groups that are exclusively specialists or generalists within the Tachinidae, and particularly within the Polideini. I have seen both cases, at least within the Tachina, for example. There are species that there are multiple records across several related species of caterpillars, as well as there are records of species of Tachina that are monophagous and are specialists. So I think that what is driving the diversification of these groups might be more associated to the oviposition strategy of this group. Something that I couldn't talk that I included in my talk because I knew I was going to run short of time. But these flies don't lay eggs. Or unincubated eggs, I mean. These flies lay incubated eggs, which are ready to emerge. And then once the female lays the egg, the fly larvae starts to crawl to find their host. So in these cases, what could be happening is that these larvae are crawling and trying to find their host as soon as they can, and in some cases they could accidentally parasitize a non-targeted host, and if they are successful and able to survive and produce and future offspring, that could be generating a host shift in those cases. I think that's what could be the main driver of diversification of these flies. Okay, so now I'm gonna turn to the audience online to see if we have any questions. Well, first with Jungle House is asking, are there other tachinic groups that parasitize these leaf-rolling caterpillars, or did this group evolve into a new host habit? It is no competition for leaf-rolling caterpillars. Yes, there are other groups of tachinids that parasitize these caterpillars, some of them with different oviposition strategies, like Celia Burrington was doing her PhD with blondelians, in which some of these species have developed this sword-like ovipositor in which they kind of slay their host and insert their egg inside their host. So they have evolved independently. Other groups. It's not an empty niche. Any other questions? Nothing here? Well, thanks to everyone for attending.